And I'm going to do that by saying welcome to another Massmedic program. Thank you so much for all of you joining us. My name is Rachel Robinson. I'm the Vice President at Massmedic. And if this is the first time that you're attending one of our programs, thank you so much for doing that. And if this is not your first time, well, it's great to see you again. We do have a wonderful program today, so we really appreciate Boston Analytical putting this together. I know it's an incredibly hot topic and something that people have really is top of mind when it comes to the medical device industry. So before we get started, I did want to take advantage of you as a captive audience. I know many of you are dealing with the back to office planning, right? So there's been a number of questions and comments we've gotten from members about what industry organizations are doing about going back to the office full time versus remote. So what I'd love to do is put up a really quick polling question. And if you'd be willing to share, we would really appreciate your input. So I'm going to go ahead and launch this poll so that we can get an idea of what industry companies are looking to do. So I'll leave that up for just a moment and then we'll move on to the program. Great, I see so many of you filling out. Thank you so much. We'll give it one more quick second and then we'll get started. Okay, 3, 2, 1. Okay, ending the poll. Thank you all so much for doing that. We really do appreciate your feedback. And with that, I am going to turn things over to Yvonne to introduce herself and Boston Analytical and to get this program started.
Thank you, Rachel. Hi, my name is Yvonne Yavar, and I work at Boston Analytical as a principal scientist for microbiome. I'm really grateful to Massmedic, obviously, for this great opportunity to present some of, our little bit of the stuff we do for medical device industry specifically. Boston Analytical is a CGMP certified lab, and so we handle a ton of regulatory testing for pharmaceuticals, biologics, and also, of course, medical devices. And so we're happy to partner with Massmedic to share some of our industry knowledge with you all. And I hope that you benefit from this presentation. Now, if you were listening closely, you heard me say I am the principal scientist for microbiome, which means I am not a medical device, principal scientist. However, I do have a little bit of knowledge and background in this, and so I hope to provide some value. Today, we're going to be talking just briefly, and I have a PhD, and I know PhDs are known to be long-winded. I'm hoping to break that, mindset, so I will not be long-winded. This should hopefully be a pretty short webinar for you, leaving plenty of time for questions and discussion. So we're going to be talking about regulatory testing of medical devices, why we do the tests we do. So basically, this particular presentation is going to focus specifically on biological testing requirements. So I wanted to kick things off by just going over what medical devices are, just so we're on the same page. So I'm using the definition given by the FDA that a medical device is used to diagnose, prevent, or treat a medical disease or condition without having any chemical action on any part of the body. So this means that medical devices are considered, non-chemical treatments or preventative devices, and they range. They're pretty diverse, and so there's diversity in the kind of product it could be, in the intended use, and the materials used, and then of course the FDA has classes for medical devices. Of course, all of y'all probably know all this information, but medical devices do go through a review process by the United States Food and Drug Administration. Just like most drugs and biologics before they can be marketed or used in patients. Obviously, given the important purpose that medical devices serve and the life-changing functions they serve in patient populations, it's very important to make sure they are safe, which is why the FDA reviews every medical device before it gets on the market. So let's talk real quick about classes of medical devices. So the FDA has 3 different classes, and they range from class 1 to class 3, an increase in what they call risk with each class. So class 1 medical devices are low risk devices such as non-electric wheelchairs or bandages or just surgical instruments. So I'm sure a lot of you are familiar with this and are in this space. And then class 2 medical devices are intermediate risk devices, just as you can see in the CT instrument right here. And then of course the pumps for IVs, those are examples of class 2 medical devices. And then class 3 medical devices are considered high risk. These would include things like the pacemaker, which is, very, very important and helps to save people's lives, literally. And so, of course, the risk associated with it is high because it is right inside of the body. And we can also talk about things like deep brain stimulators, which are implanted inside of the brain. And so, again, pretty high risk since they are in direct contact with both the cardiovascular system and cerebrospinal fluid. And so medical devices are basically regulated by the FDA, but there are multiple guidance documents. And again, this is United States specific. This presentation is geared specifically for the United States. So the specific guidance documents that relate to medical devices in the US are ISO 10993, the biological evaluation of medical devices, which actually talks about a lot of the tests that I will be describing later. Then ISO 11737 part 2, published in 2009, covers a lot of the guidelines for sterility testing. And then the USP chapter 71, 85, 1663, 1664. There's a lot of chapters in the USP that also apply to medical devices. And the reason this is in here is because a lot of the methods that we do use to do the biological evaluations are based on USP chapters. And then the Code of Federal Regulations also has several parts that relate to medical devices, like parts 800 to 1050. But of course, parts 1 through 99 are just general medical requirements, but these also apply to medical devices. And it's important to note that a lot of the biological evaluation tests I will talk about require ICH guidelines for designing the tests and for validating them. And so at Boston Analytical, we use these ICH guidelines to make sure that the tests that we do are ICH compliant. So here's a real quick spiel, very short summary of how devices basically go from I have an idea to available on the market. So let's say a scientist has an idea and it's a concept, it's developed, then that device is then designed. In a lot of cases, some of these devices will require testing in animals, especially for class 3 devices. And then that device may proceed into clinical trials. Again, this is typically class 3 devices. Class 1 and class 2 devices are typically considered, they can be exempt from review. A lot of class 1 devices are exempt from review by the FDA after submission to the FDA for approval, typically because they are considered extremely low risk, but also because a lot of those are similar to existing devices. So basically to be exempt from review, basically fast track to approval, if you will, class 1 and class 2 devices need to be extremely low risk or similar to existing devices. However, they must still comply with quality control and manufacturing standard requirements, right? So a lot of the tests I will describe will typically still be performed on some of these devices, not all, but just because we want to make sure that these devices are still really low risk, right? We have to establish that they are low risk. So non-exempt class 1 and class 2 devices, basically class 1 and class 2 devices that are considered not exempt from review, will undergo 510(k) reviews. 510(k) is a pre-market notification review process. And this process is basically to establish if the new device is substantially equivalent to an existing device. So if an existing device is available for comparison, then we're trying to, and that device has already gone through, testing and all of that. So it makes it easier to take this new device and compare it to the existing one and then establish similarity or equivalence, basically. Class 3 devices undergo pre-market approval, which is a pretty more intense, of course, given the significant risk associated with them. And typically will include clinical trial data on the safety and efficacy of the device. And so there is an equivalence work being done here. Here, purely we're reviewing, the data you do have related to the specific device to see if it's safe. And of course, if the efficacy is as it's claimed before approval is granted. So typically devices that go through 510(k) review are considered cleared. That's the language the FDA will use. And then devices that fulfill safety and effectiveness criteria through the PMA review are considered approved. And so again, language the FDA uses just to clarify that, class 3 devices are pretty similar to a lot of drugs or chemical agents in how they're reviewed. And so that's why they will use the term approved. So after FDA clearance or approval, the device can then be marketed for sale and use in the United States. There are other kinds of processes that I will not cover here. There's exemptions for humanitarian use, for example. But again, this is related very specifically to the device you are working on. So if you want any kind of clarity on that, we can, of course, touch base offline later. The FDA still monitors side effects and performance of devices after they are on the market. And so this is a part of their responsibility. Apart from just approving it and having it on the market, they will still monitor the device through reports, typically, after they're on the market. And manufacturers also have to fulfill any additional surveillance requirements, depending on the type of device that they are marketing. All right, so now I'll move on to the meat of the presentation, if you will. So I will talk about the most commonly performed biological evaluation tests that we do. So I will cover sterility testing, I'll cover extractables and leachables, even though, again, I will admit I'm not an expert in ENL by any chance. And then toxicological evaluations, which is kind of connected to ENL testing, then bioburden tests, and then endotoxin testing. And as I mentioned before, all these methods must be validated for the device under testing according to ICH guidelines. And that's typically what we do at Boston Analytical. So let's kick things off with sterility tests. So why a sterility test, someone might ask? A sterility test is basically to establish, right, that the tested product meets sterility specifications. So what are these specifications, you may ask? Basically, if that product is, for example, a class 3 device, which is high risk, obviously has to be sterile, right? We don't want any microbes on it when it's going inside of somebody's heart or brain, which can cause an immune response or result in illness, so, or infection in an already typically a patient that already has underlying health conditions. So we use a sterility method that is based on USP 71 and an ISO 11737 R2. So we offer both either direct inoculation or direct immersion of the device, where we take the device and immerse it directly in the medium to be tested, and then we culture that for 14 days to recover growth, or membrane filtration. Again, we would, in this case, filter and then wash the filter before culturing for 14 days again to try to recover growth. And in either of these cases, we try to make sure that we are following ICH guidelines, which means we're doing it in a certified clean room, and we are doing suitability and method validation. So we do bacteriostasis and fungistasis. And typically, we will use typically direct immersion with medical devices. However, in some cases, we'll use membrane filtration, especially if either bacteriostasis or phagostasis is detected, which basically means that after we immerse the device, we notice that it basically, or during suitability and validation, we notice that there is any component of that device that is impairing the growth of the microbes. And so to prevent that, we would then do a membrane filtration and wash, which will typically get rid of that inhibitory factor. So that's it for sterility testing. And then for leachables or extractables, first of all, we use a method that is based on USP 1663, right? So extractable studies expose the device or its containment or enclosure system material or the manufacturing component. So in this case, we expose the device to stress conditions, and then we extract potential contaminants from the material. And we use heat or multiple solvents and exposure time to basically establish if there is any component of this device or anything that is being extracted out of this device that would require further testing. So we will typically go ahead and ID what compounds are present after an extraction is performed. And then we do this to understand if those extracted compounds would pose potential toxicological harm. So if any of these would, we would proceed with leachables testing. So leachables testing is based on 1664, and they're designed to identify if the components identified in a extractable study will leach out of a device, first of all, in controlled conditions over time. So we perform these directly on a medical device, and the results are correlated with extractable study results typically. And then if any leachables are detected, right, if you have anything leaching out of your device, those are evaluated toxicologically, basically to make sure that they're not having toxic effects. And so it's important, of course, again, to emphasize that these methods have to be developed. There has to be suitability performed, and then we have to validate the methods. And we will typically do this specifically for the medical device because, again, remember, they are so diverse and there's so many of them. So we have to make sure the method that we are using is appropriate and giving us reliable data for the device in question. And then we will, of course, also validate these methods to target the potential leachables that were identified in the extractable study. So extractables and leachables testing is typically they're like twins. They typically go together. Okay, so bioburden testing is based typically on 7372, the ISO chapter, and also, of course, USP 61. And it quantifies the microbes that are present on the medical devices. So the sterility test is establishing that there are no microbes present, right? Whereas the bioburden test is basically attempting to quantify the microbial load on the device, especially for things like class 1 and some class 2 devices that are not really going to be completely sterile. So these require suitability testing or method validation, again, according to ICH guidelines, just to ensure that the test method will allow the growth of microbes that are present on the device. And so we will typically spike a low load of 6 different microbes that are named in USP 61, and then try to recover their growth from the device, just to make sure that the device isn't preventing the growth of the organisms of interest. So bioburden test suitability, just as I mentioned, we are spiking and then recovering using the exact same method we would use for the actual test. So after this validation is performed and we're sure that the testing conditions can give us reproducible results, we will go ahead and perform bioburden testing to determine the microbial load. So this you're plating and then you're growing out to sea. And then typically we will use this load in sterilization validations if needed, just to determine the sterilization load. Okay, so moving right along to endotoxin or pyrogen tests. And these, the name sounds, really scary or something complicated. It's actually one of our fairly simpler, stable tests. We run a lot of these. So just as a little background, endotoxins are toxins that are inside of bacterial cells that are released when the cell lyses or breaks open. And this typically happens when the bacteria dies. So the most common example of endotoxins are lipopolysaccharides. So lipopolysaccharides are components of the cell wall of gram-negative organisms like E. coli or Salmonella. So typically these lipopolysaccharides are endotoxins. So when you hear people talking about endotoxin, they will typically be referring to LPS or lipopolysaccharide just because it is the most common one, right? These endotoxins can cause very severe reactions and disease in patients, including like hemorrhagic bleeding, and then they can basically cause like an immune response that can result in shock, toxic shock. So it's very critical to ensure, especially for devices that are, again, in direct contact with any bodily fluids, that we are not exposing the body to loads of endotoxin that will result in any of these adverse reactions. And so the endotoxin test is based on USP 85. And so again, for medical devices, just because of the diversity in them, the FDA does provide specific guidance depending on the kind of device that you are using. At Boston Analytical, we use the LAL, right? This is the established method. Oops, sorry. My screen froze for just a quick moment. All right, so at Boston Analytical, we use the Limilos amiboside lysate or LAL method. This is the validated method. But recently, just a few years ago now, a new method emerged called the recombinant factor C method. And the RFC method was developed as an alternative to the LAL method because the LAL method uses horseshoe crabs as our base for cells to test. And so as a way to protect the horseshoe crab population, the RFC method was introduced. So both the RFC method can also be validated for a sample, especially if someone is very interested in using that just to protect the horseshoe crab population. So that's an option now as well. And inhibition and enhancement testing is typically performed for each test to ensure the test is sensitive enough and there are no inhibitory compounds present on the device. And then the endotoxin quantity must not exceed the threshold limit set by the guidance document. Okay, so at Boston Analytical, we do offer all three different types of LAL tests. And then the endotoxin quantity limits, again, depend on the device type. So for example, for devices that come into contact with the cerebrospinal fluid, the limit is extremely low as compared to devices that aren't really in direct contact with CSF. And so sometimes you have limits as low as 0.006 EU per device. And in some cases, you have as high as 20 EU per device. So it's important to determine that limit for your device, specifically depending on what class it is, and we're able to, of course, offer some guidance on this if that is a need. So I did say this was going to be very brief. I hope I've held up my end of the bargain, which means I'm expecting really great questions and discussion. But I would like to close out just by saying that medical devices are very diverse, and the biological characterization tests we typically perform will rely on guidance documents, but very, very heavily on expertise as well. So going with a lab that has a lot of experienced personnel can reduce the time you spend trying to figure out how to make things work for your specific device and what specific limits or guidance applies to your device as well. So in closing, I will say thank you for this opportunity. I'll take questions, and if you need to contact us, we are a CGMP compliant lab, just as I mentioned. We're registered with the FDA. We're an agile company. We're able to, implement tests really quickly. Very, very professional people. A lot of the people are way more professional than I am. So if I'm not professional enough for you, don't worry, you're okay. So you can reach out if you have any questions that I cannot answer at the moment. You can just reach out using the emails that are on our screen. With that, I'll turn it back to Rachel.
Yvonne, that was great. So we do have some questions coming in from the audience. So please, folks, pop them into the Q&A and I'll run them through. But my first question, before I get to the audience question very quickly, is, Yvonne, how often do companies come to you with a test plan in place? They know what they need to test for, and how often are you helping them develop that?
That's actually a really great question. And off the top of my head, I don't know that I can say confidently for sure. But if I just because this isn't something I've tried to get data on, but I can if I would guesstimate, I'd say some companies do come already prepared. They know exactly what they want. They usually already have methods that they just need transferred and validated with us. And some will also come in not entirely certain what they need, or they have some idea, but they aren't sure the specific methods that should be used for their device. And so we're able to do, both. It doesn't really matter so long as we're meeting requirements. We try to make sure that we offer advice if needed. Like if someone comes in with a plan and we realize that something needs to change about that plan, we will, mention it just so they know. But really, it ranges. It's really difficult to say. Yeah.
No, that makes complete sense. Okay, so we definitely have questions coming in from the audience. So I'll ask these of you, but again, if there's something that we need to follow up with other folks at your organization to answer, then we'll make a note of it and make sure we get back to the attendees with those. So first question, Yvonne, thank you so much for the great talk. This is from a small company. They've heard conflicting advice about whether the devices sent for testing should be the final design of the device or just the final materials that are used. Do you know if there is a strong preference for the final device design to be used in testing versus materials?
It depends on the specific test, actually. And so for something like extractables and leachables testing, extractables test can use just the materials. We don't actually need the final device to do extractables testing. However, for the leachable stage, you do need an actual device. And I would say that typically we would prefer to have the actual device for things like, actually for endotoxin testing, we have to do it on the actual device. So it really depends on the actual test. So I would say that reach out and, we can help answer this question more specifically for you, because it really does depend on your device and the test you need to do.
That could be why she's getting conflicting advice. I would imagine that's probably why people are saying different things.
Yes.
So absolutely reach out to you all for that one. Okay, that makes complete sense. Okay, next question. Can you give a general sense of timeline for sterility testing and bioburden testing? How long does that typically take? And is there an average sample size?
Okay, wow, that is a really good question. So I will start with sample sizing. Sample sizing is dependent on, and this is going to come up a lot. Just know a lot of things will depend on your specific device that you're testing. Some of the chapters will actually specify the sizes for some of the devices, some don't. And so what we do have is a lot of experience testing similar devices. And so we can basically go off of previous data and give you good advice and say, hey, for this device, you might want to use the size. And I will also So for timeline, we attempt to do a lot of our testing within 21 business days. However, keep in mind, so the actual test for like sterility, we have to do the spec method requires 14 days of just culture. And so already 14 out of those 21 days is already gone. So typically the timeline is short. We do have current bandwidth for new projects, just reach out. Okay.
Great.
We can give you more specific information related to how long we would need to test your device if you reach out to us.
Great. Thank you. Okay, next question. Is rabbit testing a recognized test method for endotoxin pyrogen testing?
That's a very good question. And so the FDA guidance on this is that if your device requires endotoxin or pyrogen testing, then yes, it's still actually used. And they actually recommend that if you've been using it, you can continue to use it. So that's kind of like, again, they're basing it specifically on your device and what you've been doing. So it's still a recognized test. I didn't put it in there just because not all devices are doing this test.
Great. Thank you. How is the big picture question? So if this is a little deep, how has FDA policy changed on validation of alternative sterility testing and biocompatibility methods?
Alternative sterility testing and biocompatibility methods. I'm not sure that I can answer this question adequately. So what I will say, Richard, is if you don't mind, I'm going to go look into this a little bit and then reach out to you. If I can get your email, for example, I can contact you directly and try to get a sense more of the question you're asking, just because I'm not familiar with maybe 10 years ago what the policy was versus what it is right now. And I did realize with Arun's question on rabbit testing, I didn't touch on something. Not everyone may know what the rabbit test is. It's basically, it is a recognized test method, but it's testing for different pyrogens. And so it's not just testing for the established endotoxin that I talked about, which is what is considered the major endotoxin. There are other types. So the rabbit test allows you to screen for these other types of toxins. Basically, that's the purpose of that test. Okay.
Great. That's great. And Richard, if you are interested in giving Yvonne your email, you can send her a chat direct in the chat button, not the Q&A, and she can reach out to you with more information on that. So moving on to the next question. So David is asking, would you, and I might chime in on this one too, would you have a suggestion for a clinician scientist who has a patent on a blood sampling device, but not much else? How would they take the next step and find experts to assist? I may jump in on that one very quickly and say, David, this is something that is incredibly common. It really depends on what you want to do with the idea. If you want to go be a startup and a CEO, we would absolutely at MassMedic recommend a bunch of programs that you can help take your product to the next level. You can also link up with accelerator programs, obviously law firms who have understanding on patent options, but I think you really need to figure out what you want to do with that. I mean, if you're thinking from a testing perspective, perhaps Yvonne, you might chime in on that one.
Yeah, from a testing perspective, after you've utilized a lot of these resources that Rachel has already kindly offered, I would say still reach out, especially once you get closer to needing to submit this to the FDA. Is when you would, but I would honestly reach out earlier just to get a sense of what you might need tested. But we do try to have tests done as quickly as possible to meet timelines for submissions and such. So most people will just wait until they have an actual product that they're ready to take to market. And then they will come to us, and then we would typically offer advice on what they need. Again, as I mentioned, it varies, but a lot of people are coming in with testing plans, some are not. So just as you get closer to market is when you would need to contact us, but utilize MassMedic. They are pretty awesome.
Thanks for that. Okay, next question. This is regarding a pacemaker. So a pacemaker is implanted under the skin, close proximity to the heart. For extractables test, what kind of environment would be used to test for toxicological harm? So what kind of simulated environment is used in that testing?
I would have to say I have to get back to you on this one because my sense of that method is that it's still heavily dependent on, and I don't know if your question is directly related to pacemakers in general or related to other devices. And so I would just say that the environment we use is heavily dependent on the device. And so I will have to get back to you on specifically the environment for pacemaker because I don't have it off the top of my head.
So Mark did put another note in the questions done. Maybe this helps. So even if we talk about how we generally simulate being in the human body, do you have any thoughts on that?
Okay.
Yeah.
So you are more interested in a general simulation of, you know, the solutions, the fluids, and all of that. Mark, why don't you reach out to me after this and let me provide you with very specific answers. Just because I am not in ENL, I don't want to give you information that would be false. Like I have to be extremely careful. So my understanding of this is pretty basic, and I really don't want to provide you with false info. So I can put you in touch directly with our ENL director, who is awesome, and he can give you like very, very detailed specifics on the methods they're using if that's something that interests you.
Great. And Mark, if you also want to just send that over, you can send it over in the chat directly to the panelists, and then we'll get your email address so that you can follow up as well. Okay, let's see if I think we have one or two more. Okay, so this is a good question, but I'm not sure if it's a testing question, but yeah, absolutely chime in. How can devices be exempt from review in the classification process? This is from a small startup bringing a med device to market. I mean, I think this is a bigger FDA question, but do you have any initial thoughts, Yvonne?
My initial thoughts are that the exempt criteria are typically determined by the FDA. So to determine if your device will be exempt from review, you would have to talk to them directly. They can then review the data you give them on your device, and then they will tell you if you need to do the 510K or class 3. I'm assuming this is not a class 3 device just based on the way the question is phrased. So for class 1, class 2, typically if it's class 1, a lot of class 1 devices are exempt, but some are not. So really depends on what the FDA decides in your case based on what information you give them. So I would reach out directly to them.
Yeah, that's a great comment. And Barbara, I would say too, we did a 4-hour FDA webinar last week that covered a ton of topics, including exempt products. I can send you the video for that. The other thing I would say is, if you, as Yvonne mentioned, if you haven't talked to the FDA yet, right, pre-sub meetings, pre-cert meetings, right, these are important things to have. But you want to go in armed with some information, right? So the best thing you can do is not go to FDA and say, how do I do this? The best thing you can do is spend a little time researching, and there are FDA consultants and regulatory consultants who can help with this, where you can actually use the information that the FDA has made public on certain types of products that are already exempt that might be similar to what you're trying to do. And if you go into that first meeting armed with that information, you will be more likely to come out with a result that you're looking for. Now, they may still push back and say you're absolutely not exempt, and these are the reasons why, and this is what you have to do. But if you do a little bit of research ahead of time and come up with what you think are good example products that are similar to yours, you can go in armed with that information and have a very strong conversation with them about why you think you should be exempt, if that's the case. So hopefully that helps. But Barbara, if you're interested in that video, just shoot me a note in the chat and I can send you that webinar as well, because that was a really great FDA program.
Yeah. That's
okay,
awesome.
Let's see. I think we've just got one or two more. Is it common for medical devices to have sterility tests done for release? The medical device claiming sterile is normally achieved through validated sterilization process with SAL 10.6.
So I will say that the broad answer to this is that even after method has been, sterilization method has been validated for a particular product, there still has to be routine device sterility testing. So these routine sterility bioburden has to be done routinely. A lot of these will require like a quarterly audit, and then environmental monitoring, obviously, and all of that. So I would say just because since it's supposed to be a routine basis, I wouldn't call it like release testing. However, the regularity of it, I think, is going to depend quite heavily on how often you're having your audits and all of that stuff. So I can also say I will check in a little bit more on it, on your specific sterilization process, and then I can provide a more specific answer for that specific one. So shoot me an email. My email is on the screen. Just shoot me an email and I can try to provide a very specific answer for you.
Great. You're a rock star here. We've got, okay, just one or two more and then we're good. Are you able to perform biocomp testing or have toxicologists that can help identify what tests should be performed?
So we do have pretty experienced staff, and so they can help you to identify which tests should be performed. For biocompatibility, I will have to check and see if we're offering this. I am not particularly confident. I'll check in with the team right after this, and then I will know.
Okay, let's see. Okay, I think we got one more. That's it. Oh, we're doing so good. Okay, if a product is designed to comply, and there's some things here, with ROHS and REACH, is there a lower likelihood of finding anything of concern in an intractable or leachable test?
That's actually a really good question.
And also,
yeah. Arun is asking really good questions, but it's also really difficult to answer just because the results should obviously we should hopefully I think your guesstimate is right that there is a lower likelihood. However, because manufacturing processes change and a lot of things can vary between what you tested previously and then when we are running the test, it could be different. So I honestly I can't say. It's very, very difficult to say, 100% of the time, yes, there will be, nothing of concern. I don't think that's a realistic answer. I think just because of the variability that occurs, also between, different types of tests, I would say just, get it done and then I suppose.
Okay, one last one that just came in, and then we'll say we're good. Are there metals that are least likely to have leaching and extractability issues? So how often? I mean, do you guys advise on materials or
materials for composition? Yeah. We actually typically do not because this is
it didn't seem like that might be something in your wheelhouse.
Yeah, it's not. However, I will say that I believe the FDA has some information on this.
Yeah.
They do have some materials that, typically have less likelihood of leaching and extractability issues. So I would check in on the resources they have on their website. But I don't know if the webinar they did last week also covered some of this. Rachel is nodding.
No, so we didn't really cover materials. It's not something, the people I've actually found who are really good at material suggestions are the folks in product development. Okay. So if you reach out to product development teams, there are a number within our membership that provide great service around that. And I could suggest if you'd like to shoot me a note. They are often designing and developing a ton of different types of projects and products. So they have a very good grasp of what materials are, you know, safe and good for certain types of environments. So that might be a good route, but also absolutely take advantage of the FDA documentation to try to understand that. And if you are a large company, it's anonymous, so I'm not sure who you are, but if you're a larger firm, you may have more opportunities to connect with other product development folks who are working on that. But engineers and designers tend to have a pretty good grasp of this. So that would be my suggestion. And I think that's it for questions.
Yay, that's great. Thank you.
So first of all, Yvonne, thank you so much for doing this. For those of you that hung out with us the whole time and stayed through the questions, Boston Analytical will be doing more programs like this with us. So keep your eyes open. I know this was incredibly informative, and I hope you all got as much out of it as I did. So thank you so much, Yvonne, for doing this.
Thank you, Rachel, for having me.
Of course. And everyone have a wonderful rest of your day. We are videoing this program. We will be sending it out. And if you have more questions, Rachel@massmedic.com, and you've got the folks from Boston Analytical on the screen email in front of you. So, and those of you that sent over your individual notes, we'll make sure that we get those over to you with answers. So have a great rest of your day, everyone.